Seminar. It's a great pleasure to introduce Dr. Julie Mitchell. She got her PhD in mathematics at Berkeley in 1998. The PhD advisor was Maurice Hirsch, and she specialized in dynamical systems. And from dynamical systems, she decided to switch to computer applications and biosciences and biochemistry, and worked in the beginning at the University of California, San Diego. And from there, she got a position at as a tenured professor at the University of Wisconsin, Madison. About 6 years ago, she decided to move to Oak Ridge National Lab, and she directs the bioscience division there, which has about 150 people. And she's interested in biosciences and also in computational modeling. Last year, she participated in the Oppenheimer Fellowship, and she's also interested in public policy.
Yes.
And she's going to talk about navigating interdisciplinary research as a mathematician.
Right. Well, thank you so much, and thanks for inviting me. I always enjoy giving these types of talks. So I did have a slide about my background, which summarized quite well. But so I switched out of pure mathematics after graduate school and went to the San Diego Supercomputer Center, where I did computational molecular biology, molecular biophysics, and then was a biochemistry professor and a math professor at Wisconsin. So I had a 50-50 split tenure home, and chose to embed myself in a primarily experimental biochemistry department in order to kind of advance my knowledge of science and be more impactful in the types of things that I can do. And then around 6 years ago, I was approached for a leadership opportunity in the national lab system. And, you get to points in your career where you're like, what do I want to do next? Like, you reached wherever you wanted to get to and have kind of itchy feet and want to kind of rise up. And this was a neat opportunity. And so I went there 6 years ago, first for about a little over a year as the deputy director for biosciences, and now for the last roughly 5 years as the director. So that's just a little bit about me. So my talk is going to be 3 parts. The first part, I'm going to talk about being an interdisciplinary mathematician, just some broad advice. Second part will be opportunities in the national laboratory system. So, some of you are interested, if you're attending this seminar, you're probably at least interested in careers outside of academia. And in addition to industry careers, there are interesting opportunities at the national laboratories. And then the third part of my talk, I'll actually show you some vignettes of projects that I've worked on throughout my career where some of my mathematical thinking has been useful. So kind of 3 parts to the talk, and hopefully you'll enjoy it. So the first part of my talk is working across disciplines. So the next slide I'm going to show you is kind of a flow chart that, maybe if you're wondering what field of science could I apply mathematics and what are my interests, maybe this will help you, or, how mathematics relates to, to other fields. Some of you may have seen this. It'll give you a minute with it if you haven't seen this, but find yourself on here, take a minute. I love this slide. So, basically things, what do you like, right? But for you, those of you who are mathematicians, you're over here getting to be a fussy pedant, right? So, and you can see biology over here, curiosity, and so on. But different things. Computer science is indirect evil, and so anyway, it's a very funny slide. And so anyway, so the first slide I have here is don't be a fussy pedant. If you want to be successful as an interdisciplinary mathematician, you need to learn to work with people in other fields. And so, ultimately the point is communication. So to give you an example, say from my own career, I work with a lot of chemists, and in different ways, sometimes I've used convolution integrals. And so we mathematicians and physicists as well know that the verb form of convolution is convolve. But if you talk to a chemist, they say convolute. And I'll tell you that every single time I hear that, it grates on me a tiny little bit, but I don't correct anyone, right? Because it doesn't matter. It still bugs me because I know it's incorrect, but I'm just not going to be that person, and neither should you. If you want to work with chemists, they're going to say convolute, and you'll just have to live with it. So that's the first piece of advice. The second piece of advice is learn to explain things conceptually without excess detail. Do you know how to explain what you do to a 5-year-old? I work on molecular docking applications. It's like 3-dimensional puzzle pieces, right? Like how can you use common objects in order to explain what you do, right? In ways that are accessible to everybody. And that's an important skill if you want to work across disciplines, to be able to distill things down. And also nobody cares about your theorem. It's fine if you do theorems and proofs, but nobody cares about your theorem. Most people don't care about your equation either. They just care what you can do with it. And so understanding that, when I was a biochemistry professor, we would go, it's a little different than math. There's kind of a little dog and pony show that happens every year where the faculty get up and present what they do and try to recruit the new students right into their programs. And, the first 2 years that I did this, I was putting up partial differential equations and they looked like I had hit them with a truck. So you need to know who your audience is and what's going to appeal to them. Once you get rid of maybe some equations or simplify things, it'll be a little bit easier to work with other people. Don't bring a hammer looking for nails. Is the third piece of advice. So, we all have mathematical techniques that we like. We have different types of codes and so on that we're interested in. But, I was in computational mathematical biology, at least the kind of modern version of math biology fairly early. And so, when I went in, people were like, what are you doing? And then 3 years later, they're like, hey, how can I get into this? But I'll never forget, there was a man who came to me. He was a differential geometer, and he says, I would like to use fiber bundles to solve the protein folding problem. And I said, okay, but he didn't know anything about the protein folding problem. Literally didn't know any of the physics, didn't know the existing literature, didn't know anything about how people had approached it in the past, but decided that fiber bundles would somehow be the solution to this, right? And so you don't want to do that, right? It's okay to have techniques that are at your disposal, look around for compatible problems, but you can't just decide that the math that you love automatically applies to whatever problem that captivated you in science. And then of course, be curious, learn some science. So as I mentioned, I chose to be 50% faculty in a biochemistry department to embed myself in a primarily experimental biochemistry department to make sure that the research that I was doing was really relevant to science and to have more of an impact. And so, so anyway, that's kind of just some, some simple advice on, things that I learned in this, in this trajectory that I think are helpful in terms of thinking about, working across disciplines. Okay, so now let's hop to the second part of my talk, which are the kinds of opportunities at Oak Ridge or other national laboratories. So there are 17 Department of Energy national laboratories. I think 4 of them are what they call NNSA labs. So those are like more like the weapons labs, the defense labs, like Los Alamos or Sandia would be examples of those. Actually, Los Alamos is Office of Science, but Sandia is in the national security space. And then there are Office of Science labs, and most of them are devoted to clean energy applications. So in my division, for example, we have a lot of people that are working on sustainable aviation fuels. Other people work on battery technologies, different types of materials, high performance computing, right? You know, we have a neutron science facility where they have, you know, beam lines and look at, you know, the structures of materials and molecules and things like that. And so those are, you know, there are 17 of those national laboratories that have kind of different emphases. Oak Ridge is a lot on the high performance computing side, and then the neutron science side. Those are kind of two of the signature things. And we also have a pretty big biology program. Actually, my division is the largest research division in terms of research dollars. So, and mathematicians, so we have a math and computing directorate that employs a lot of people, you know, that do all kinds of applied mathematics. And then there are mathematicians that are embedded within the more experimental programs. And so there are different kinds of things you can do. And again, I think I kind of touched on this a little bit, but you know, kind of some strengths. Materials, computing, also isotopes. So Oak Ridge makes the medical isotopes that go into cancer treatments and tracers and things like that. So is, I think, the world's largest producer of isotopes that are for medical application. Biological environmental sciences is also obviously, you know, where I am. And then we have like a 3D printing. I mean, there's all kinds of different things that you can get into that are really cool. You know, and then looking toward the future, you know, we're looking at carbon negative technologies, you know, biomanufacturing, like, you know, how do we do clean, you know, kind of clean biomanufacturing, you know, sustainable aviation fuels I mentioned, and so on. So kind of what's the future of energy research? Or there are other types of things that people work on as well. I mean, we all hear things about, say, you know, outside of my division, we've all heard about we need grid modernization for energy. Well, you know, underlying thinking about, you know, grid modernization and how you distribute energy through systems, there are mathematicians working on, like, you know, network, right, you know, kind of network models for how all that works, or machine learning models to kind of pinpoint, like, hey, we're having a problem right here, right now, and here's what we need to do about it. And so there are different types of opportunities at Oak Ridge and other national laboratories for mathematicians doing really interesting types of science. And in contrast to, you know, I mean, I always think of, you know, at least my own graduate experience was, you know, sitting there with I mean, I was a pure mathematician. So we had, like, you know, pencils and paper and sitting in your office by yourself, like scribbling stuff on paper. The kind of work style at a national laboratory is also really different. So it's big team science. And so you won't necessarily be working on, you know, a highly individualized project as much as things that allow you to collaborate and work across disciplines, you know, and work with other laboratories, universities, different things. So lots of different things you might do. In terms of what kinds of opportunities there are for people, we do have programs. Many of you are graduate students, so we have you know, we do have graduate programs and internships. Postdocs, if you guys, even if you think you might want to go into industry or you're not sure or whatever, National Lab postdocs are great. They're really interesting. And also they pay, like, you know, we're paying like $75,000 for the postdocs per year. You know, that would be your salary in my division. And I think the computing people are making, you know, more like 85 or 90. So it's like a pretty good salary compared to some academic salaries. And we'll give you a taste and a skill set that's maybe closer, again, even if you're interested in industry, your skill set will be closer to what industry is looking for, because industry is also looking for people who can do these kinds of interdisciplinary team science type activities. There are visiting faculty opportunities. Some of the paid programs to the Department of Energy are mainly for, for faculty that are coming from minority serving institutions, but we can also arrange for faculty to come as guests for some period of time and work with us. Undergraduate programs, internships, and then some programs that we have, kind of gap year programs. If somebody is, they want to take a year between after their bachelor's degree before going on to grad school and maybe do, work a bit, or after their master's before their PhD. So we do have ways to set you up with longer term paid internships. So that's, some of what we do. And these are some specific programs. So you can find, so SULI is from the Department of Energy paid programs. And then URSI and GERSI, so that's undergraduate research something internship and graduate research something internship. I don't remember what the S stands for, but those are programs, like if you identify a project, somebody leading a project that you're interested in that you've been talking to, so those would be out of our project funds. And then the SULI program is specifically for undergraduates, and that's paid through the Department of Energy. The GEM program is for underrepresented students, graduate students, so really taking people from some of the best universities around the country and giving them a summer research experience. So this year, my division hosted 8 GEM students, which I think was actually a third of all the ones that came to Oak Ridge, which was really fantastic. And then again, I mentioned the visiting faculty program. That's a Department of Energy program for, if you know anybody at MSIs that's interested in coming, and I hosted somebody, to come basically and work on a machine learning project. And the idea is, he's taking this skill set and knowledge back to his university, right, in Fayetteville and North Carolina, and, bringing that back to his students. So, and then what kind of careers are available? There's not just, sometimes we think, when we think about research, we think of, something like a faculty job. So the closest that we have to a faculty job at the National Lab System would be a staff scientist track. And so there's not really a notion of tenure, but there's definitely like an advancement through levels. So we have 6 levels of staff. We would, if you have a PhD, we would hire you at level 2. Level 5 is the highest most people get. And then we have what are called our corporate fellows, which would be sort of, like the really distinguished professors or endowed chair type people, like that would be our level 6 folks. But, so these are, these are folks that most people will be working with a smaller group. So they might have 2, 3, 4 postdocs, something at that scale where maybe they're managing, I mean, when you're starting, you're kind of hustling, and, it takes a little while before you're maybe you got your first postdoc or something like that. But by the time you've risen up the ranks, maybe most people are, managing a small team, some postdocs, technical staff, and so on. And then you still have some people that are managing even larger projects. So the largest project in my division is $27.5 million project. And so we do have somebody that's the center director for that. So there's sort of, a range. Now, there are additional tracks. So if you want to be a staff scientist, and the national labs are what we call, and maybe you guys are graduate students, so you may not know this terminology yet, but soft money positions, in the sense that you're completely supported on grant funds. So when you're a professor, your salary, at least for usually 9 months out of the year, is covered by the university, and maybe you come up with some grants for summer salary. In the national laboratory system, all of your money comes from grants. And before you think, oh, that sounds like an absolute disaster, I mean, the Department of Energy, you know, like we work with them to make sure we have enough money to cover people and so on. So, you know, everybody's a little stressed about it once a year, like, where's my money coming from next year? But at the end of the day, everybody ends up fully covered for the most part. But that's, you know, that's the kind of staff scientist job. But there are additional tracks where you're not responsible for bringing in the money. So one of those would be a technical professional track. And these are, you know, if you even have a master's degree in math or computing, you could follow this track. Some people do it at PhD level. A little more unusual maybe in math or computing to have somebody come in with bachelor's, but it's not impossible if you're good just to come in with a bachelor's degree. But you know, basically you would be working in support of the staff scientists, you know, maybe one or more staff scientists cranking out codes, right? And, you know, getting data and results that they need for papers. And those aren't going to be paid as well as the staff scientist positions, but they're still, you know, they're still decent, good jobs. And at Oak Ridge, we have a pension system. So, you know, it's you know, it can be a good, you know, kind of a good solid career track. And then as you know, if you want to go into some leadership, there are opportunities to be a group leader. So then you have some what we call line management responsibilities where you know, you hold performance evaluations with people, you know, help with some administrative things. My job is division director. So then you have, you know, more significant administrative responsibilities along with strategic planning, relationship management with the Department of Energy, things like that. And there's still some opportunity to do research. At that point, it's a little bit up to your choice how much you want to do, if you want to work 80 hours a week or 45. So, you know, and then for, you know, maybe a few of you, there are careers in around technology transfer. So some people go back and they'll do some, you know, maybe do law school or something related to that and actually do, you know, something that's a hybrid between science and law in order to understand kind of, you know, help people get patents on their research and do technology transfer work with companies to get things licensed out into the community. So anyway, those are some potential career paths for people in the national laboratory system. And again, you know, like the staff scientist is closest to being a professor, and say the division director is closest to being maybe a dean. There's not, I don't know that there's anything that's exactly like a department chair, but you know, those are the closest mappings. So, and then I'll spend the rest of my time just talking about some of the projects that I've worked on, starting from, you know, very early in my career in interdisciplinary science, and just to show you, you know, some mathematical thinking and how you, I don't know, just, you know, ways that I've at least contributed, and then you'll all find your own paths. So first I'll give you a very quick molecular biology lesson. So we all know about the DNA double helix, right? DNA encodes RNA. RNA through the ribosome encodes for proteins. Proteins fold up into characteristic shapes, right? So, you know, some people are now familiar with, you know, protein folding is making a resurgence due to AlphaFold. So people are interested in the protein folding problem. And then the area that I've spent most of my time working on is not on the folding question, but after you have the proteins that have folded up, how do they assemble themselves? And so, and this is how all the physiology in your body happens. You know, things bind and unbind. You know, a drug binds to a receptor, or an enzyme binds to an inhibitor that stops that reaction, right? So, different types of chemical reactions that happen. And so I've always started out working on the protein docking problem. And so you can think about that as, think about them first as rigid bodies. They're really flexible and dynamic, but you can hold one fixed, rotate and translate the other, and try to crash them into each other. And so that's molecular docking. Like I said earlier, it's a little bit like three-dimensional puzzle pieces, but then there's some chemistry and physics on the surface that you also have to account for. So what types of problems, have I been able to work on? So electrostatics is a cool and interesting problem. So you have proteins in solution, and the solution has water molecules and ions. So think about it as basically like, water and salt. And what happens is the protein surface has electrostatic forces, and the water molecules will align themselves. So the water molecules have a dipole moment. The oxygen it pulls electrons, so it's a little bit negatively charged, and then you have two hydrogens. So in terms of partial charges, they end up a little bit positively charged. And what happens is the water molecules, if you have a positively charged surface, the oxygen wants to point at it, right? Because you want the positive charge likes the negative charge. And so you have this polarization effect, which is basically that epsilon in the first part of the equation is a dielectric constant that accounts for that phenomenon. In addition, you can have ions. So this particular version of the equation is for sodium and chloride. So they both have +1 charges, or +1 and -1 charges. The full equation could also have magnesium and other things, but this is kind of the simple version with just water and salt. And so the positively charged ions will want to accumulate in the negatively charged regions of the protein, and vice versa. And so you end up with this nonlinear partial differential equation that you can also then linearize, and you can see Yukawa potentials that would actually solve the linearized version. If you replace the hyperbolic sign u with just u, you'd have the linearized version, and this is sort of the point potential for that. And so using that as part of potential energies, so one of the ways you can approach molecular docking as an energy minimization problem, and I'll show you a little bit about that on the next slide. Trying to minimize potential energies, and you can use this kind of PDE formulation to characterize the electrostatic component of that. There are other components. So the first project that I worked on, that was my own thing, was the docking mesh evaluator, and it was basically set up to run on the supercomputers. So would distribute a bunch of local minimization jobs across the supercomputers, with the idea of doing global optimization. And so you have a potential energy. The assumption, which is of course wrong because we don't know Mother Nature's energy function, which is why I started trying other things, but you can do a global optimization to get to the global minimum of energy, and this is challenging because it's a highly non-convex energy function, so you have lots of local minima. But I worked on some methods like underestimator methods that basically could find from the positions of local minima what the global minima was. And so and then you can just kind of see here, so this is a trace of a protein. The colored dots represent positions where there's a good fit, a good docking position. And then the colors, red means the energy is lower, so those are better. So here you can just kind of see where you had from kind of a uniform sample, and then you do the optimization. So you have like the local minima that start to cluster in the correct location. But still, you know, global energy minimization was not necessarily always the best approach. But allowed me to do some fun things, thinking about coordinate models. And so when I mentioned protein docking, I said, well, you can shift it in 3 dimensions and rotate in 3 dimensions. So that's, you know, that's R3 cross SO3, but there are different ways you can decompose that space. And so if you think about what you're doing, docking 2 things together, it makes more sense to figure out which 2 faces, right? Which 2 faces of the proteins are facing each other, how far apart are they, and then there's like 1 additional twist dimension. And so thinking about different kinds of coordinate models and being able to digest that in ways that were useful to solve optimization problems. And then one of my most cited papers, and it's so fun to see where this paper was cited, because I've had citations from like people who work on you know, like radars and stuff like that, right? And, you know, a lot in my space, which is, you know, molecular docking, but like just all kinds, all across science, this work gets cited in different applications. So it's really cool to look at the citations. But how do you come up with a uniform sampling of rotation space? So, I mean, you know, some people think about rotations in terms of quaternions and different things. Some people think about Euler angles. If you just do uniform sampling of Euler angles, that's not a uniform sample, right? A uniformly distributed sample. And so how do you do that? And so came up with a method. I think it's the only paper that, the only papers that I published as a math professor that had theorems and proofs for around this work, kind of proving that it worked. But basically the strategy, and I did this not for SO3, but all of SOM. So you can just kind of keep going. But you can kind of think of SO3 as almost like it's not a product space of a sphere and a circle, but it's almost like a twisted product. And there's something called the Hopf vibration for those of you who are interested in dynamical systems and geometry. And so, you know, you can kind of think about this, but the easy way to think about this is how would you select a rotation? So, you know, I'll put my figure like this. Where does the z-axis go? Follow the geodesic. Now there's a turn, right? So I've reduced it to a sphere and a circle. Right? But if you want to do that uniformly, you have to figure out how to balance. If you want a sample that traverses a space, a discrete sample, you have to pick the number of points on the sphere, distribute them uniformly, and pick the number of points on the circle, distribute those uniformly. And when they combine, it has to be uniform in the 3-dimensional space. And so again, that was a fun paper that I worked on, and it's been yeah, again, it's been really fun to see how people have used it. I mentioned convolution integrals a little bit earlier. And so the first time playing with convolution integrals in this space was around atomic density methods. And so and I started this work with a postdoctoral advisor. And so the idea is how do you determine the shape of a protein? How would you pick out the crevices, the protrusions, right? Because it's, you know, if you have a smooth surface, something, you know, that looks here for the online people. So if you have a smooth surface, so something that, you know, you can go ahead and do principal curvatures or whatever, but those aren't particularly useful. If you have a molecular surface, the principal curvatures are either going to be like based on the atom that it's sitting right there, which might be a carbon or an oxygen or whatever, right? Or it's going to be some saddle-like shape. And so gives you really no information about, you know, what kind of a local environment you have. But there's a little game you can play And here up in the graph, you can see it in, like the two-dimensional version of this, which is to pick a point near the surface of the protein. And then, so in biophysics, we often use angstroms as a unit of measurement, so 10 nanometers. And so, we, play a little game. How many atoms are within 1 angstrom, 2 angstroms, 3 angstroms, up to 10? And then graph it, right? Make a little plot. If you're in a crevice and you're surrounded by atoms, that's going to shoot up more quickly. If you're near a protrusion, it grows more slowly. If you're near a flat surface, you'll have something in between, right? And you can do some calculation that's sort of, how do I want to say, inspired by a fractal dimension, but it's just kind of a logarithm. So you just kind of look at, how quickly things grow according to that radius, and use that lambda. So, grows as r to the lambda, use that lambda as kind of like a parameter that tells you something about the shape. And then if you want to do all this quickly, rather than counting the atoms, well, what you can do is you can put ones and zeros out on a grid, right, according to where your protein inhabits the space. And then you can make discrete balls of various sizes and take a convolution integral of those. It's really a correlation integral because you have symmetry. It's the same thing, right? But, do a convolution or a correlation integral. And for the entire 3D grid, you can get these numbers in one go, right? So anyway, so that was a fun project. Now, where did that lead me? And when I was interviewing for my position in Wisconsin, I gave a talk to the math people, I gave a talk to the biochemistry people, and I showed biochemistry people this, and I was very interested and had done just a tiny bit of work because I was interested in molecular docking. So you have the crevices and protrusions, but what happens when they match across an interface? And understanding the flat surfaces, when you have molecules that recognize each other, right? Flat surface doesn't tell you where to bind, right? But when you have crevices and protrusions matched together, they lock. And I was interested in that, as well as what happens when you do mutations, which those of you who know biology, mutations are really in DNA and side chain substitutions happen in proteins. But anyway, we often call them mutations, and that's what we mean. Like, what happens if you change the protein in a particular spot, and how does that, can you go back and look at evolutionary data and kind of figure out how these things have been optimized or so on. So, I presented that, and I'm on 2 patents for, I can't remember why it didn't get to phase 3 clinical trials, but a cancer drug that was based on this idea. So you have the green molecule there is a ribonuclease. Oh, excuse me. Yes, the green molecule is a ribonuclease, and the purple molecule is its inhibitor. And so a ribonuclease can chew up RNA. That's its job. The inhibitor keeps it from chewing up all your RNA all the time, and that's its job. Now, in cancer, you would necessarily want to shut down things like cell division, right? That's also how, chemo works and so on, is by, targeting rapidly dividing cells. But if you make a ribonuclease that's a super enzyme, right, that doesn't bind the inhibitor, you can get it on overdrive so that it's going to do essentially what chemo does, but in a way that's not going to make you sick. And so we did that based on these shape markers, and did 4 mutations in the naturally occurring enzyme, and in that way was able to actually make a cancer drug that was based on a protein. And again, I forget why it fell out of clinical trials. It made it past the first phase, but I don't quite remember. But anyway, and then going forward into the future, and this is where I started to get into machine learning, because I was interested in understanding this problem of what happens when you do mutations in the molecules? What does that effect does that have on association between the molecules? And so came up with machine learning models that for what we call hotspots. And so we measure binding energy so the energy of the protein by itself is G, right? And then when the change in energy for a binding event is delta G, and the change in energy as the result of the mutation is delta G, right? So delta G would be like the energy change, the binding free energy change associated to that mutation event. And if it's at least 2 kilocalories per mole, so good energy is negative, right? That's when things associate. Positive energy, right? So having the energy go up means they associate less tightly. But you basically have a few of these spots that hold the protein complex together. And can you use machine learning combined with descriptors, features that are based on biophysics that and be able to use machine learning to pick those out? And so we were really, we did kind of the first machine learning model for this type of work. And so I think we started that in around 2005, I think. Yeah, so fairly early, and my very first graduate student in biophysics worked on this problem with me and became kind of the signature for my research is this machine learning in biophysics, well before AlphaFold and so on. Not that we took exactly the same approach as AlphaFold, but we were definitely using a lot of machine learning very, very early on and applied it to all kinds of different things. So another type of problem we worked on, DNA binding site prediction. So I mentioned electrostatics before, very early in the talk, but coming up with all kinds of different biophysical features or physicochemical features, whatever word you prefer, to figure out sites on the surfaces of proteins that would recognize nucleic acids. Which seems like it would be a simple problem. DNA is negatively charged, just look for a positively charged patch, but it's actually not that simple because you can have things that look like a positively charged patch from here, but when you get up close, it's not that way. And kind of looking at local neighborhood environments, again, employing things like convolution integrals to capture information from a spatial region and map it into a descriptor that then you could feed into a machine learning model and make predictions about the sites. Okay, so I told you not to have a hammer going and looking for nails. But once in a while, serendipity happens and you happen to see something so in this case, my docking community runs these kind of community assessments where you get problems to work on and you have to make predictions, and then and then whoever does well, gets a pat on the head. And so anyway, one of these was for heparin binding to a protein. And so, I looked at the structure of heparin and I said, that looks like a phosphate backbone from DNA, right? It has some chemical similarities to a phosphate backbone of DNA. And I had this new toy, which was a binding site identifier for DNA. So ran it through, the region here that's sort of all lit up in cyan and green is where it said the heparin should bind. The predictions we made, were these over here. So I think there might be 5 predictions or something mixed in there with the chemical structure of heparin. And I guess I didn't put the slide where you can see, but the heparin binds right there, right? So the site identifier nailed it exactly, which was pretty exciting for us. Another type of problem that I worked on was allostery, which is a phenomenon where you have a binding site at one part of your protein, and it causes a conformational or a shape change in another part of your protein. And to figure out, you can imagine that mutations at either site would impact this, but there can be mutations in the middle of the protein that aren't connected to either site that can impact this. And so we did a machine learning model along with some, you wouldn't even call it explainable AI, but some type of analysis to figure out what was going on. And at least our hypothesis based on the results was that it's very mechanical in the sense that we had a parameter, one of our descriptors, that had to do with local structural entropy, or basically it would be how deformable the structure was. So that if you had a helix that was very rigid between the two sites and you pushed here, it would push there, right? And if you had something someplace in the middle with more structural entropy, it could buckle and absorb that change. And so, coming up with ways of descriptions. Let's see, what time? It's 10 past now. What?
Right, so it's good to stop soon so we have time for questions.
Okay, yeah, that's why I'm asking. Okay, I'll just hustle. This was the first particle code for graphics processors. So we did I was very enamored with GPUs when NVIDIA made their announcement of CUDA, and I was like, let's do something right now. And so we did, basically a salvation model using GPUs and published that in, I think, 2009. I'd have to double check. There's the speed ups, it worked. Let's see, probability scoring at hotspots, looking at kind of coupling in the sequence distribution over the course of evolution between two amino acids and trying to figure out whether or not they would be proximal in space. And this is actually part of how AlphaFold works, although our strategy was a little bit different from that, but use that same type of idea to do protein docking. And then the latest sort of thing I'm working on is generative adversarial networks for designing protein structure. And so here, here's just kind of a simple hairpin structure, but how can you make a machine learning model that will actually be able to design things, right, according to some prescription. And so, basically, for those of you who don't know how GANs works, there's a discriminator. It's actually how deepfake imagery is created, right? Models for that. So, there's a discriminator model, and it says this is real or this is fake, right? So it just has two choices, real, fake, and then it tells you, like, go that way in parameter space, right? And then you sort of figure out how to map that back into something that corresponds to your physical problem, and so on. But then there's a generator, right, that generates the designs. And so they go back and forth and they work together until you get some convergence, until the generator can fool the discriminator. And so anyway, we're using that. Oh, I see. I have a. Yeah, we've been using that to, again, design some of these small peptides with the idea of doing things like designing small biosensors that can change between structures according to a chemical modification is kind of the next thing we're working on. And we were being cute, so it's a beta GAN, so began. But our original name for it was, so Linus Pauling is the person who originally discovered the fundamental structures, the alpha helices and beta sheets in proteins. So we called it Ganus Pauling, but that was being a little too cute. So we came up with something a little less so. And anyway, that's kind of where I am right now with things. And that's my last slide. And then my very last slide is, please contact me. That's my email if you have questions, if you want to want to chat about research, or hey, will you look at my CV or something like that and give me advice, you know, you're definitely welcome to contact me, and you know, I can hook you up with internships or, you know, whatever interests you. So anyway, that's what I brought to talk about. And I 2:15 exactly. So anyway, but thank you so much for, you know, for being here and for inviting me, and I'm happy to take any questions.
Thanks a lot. I also want to tell the people at home that they can type the questions in the questions and answer box. Let's see. So until people think about questions, I'm curious. So for vegans, so you're trying to create things with a certain structure. What about generating things with a certain function?
That's harder. Yeah, I mean, I think the next level will be that, and structure and function are related, but you know, I do think that maybe the next level of generative models would, you know, will be designing a little bit more for function, but that's a harder question, and we don't even know, how do I want to say, we don't even know for the naturally occurring existing structures in nature how to always map that to function. And that's probably, and there are machine learning models and more for that. So but in some ways we might need to solve that problem somewhat first before we can then design for it, because we don't totally understand, you know, we don't totally understand how to go from the existing structures to the function always.
But does anyone try to do it? Because
oh, sure. Well, I mean, at Oak Ridge, Ada Sedova did across an entire genome, right? And really trying to assign function to things. And there are other people as well who really, there are like, they call them EC numbers, and it's just basically an assignment of function for enzymes and other things. And so, yeah, like how can you make a machine learning model where you plug in the sequence or you plug in the structure and it's going to spit out an EC number? So there definitely is progress in that area, but I think there's probably a ways to go.
what about the stability of the results? Because they can be very sensitive to the initial parameters, right? So
do you mean the GAN models, or do you mean
the generator?
Right. We didn't run into too many problems. I mean, we didn't have any mode collapse, and things, you know, seemed to converge pretty well. So, you know, maybe we were just lucky. I mean, this, you know, but I want to say at the end of the day, you get kind of a distribution in space, so that, you know, the generator basically says, you know, pick things over here, and you get a vector of numbers, and you have to figure out how to make that a protein sequence again, and so on. So there are, you know, there are different challenges, but we never, you know, we didn't have any issues. And when we used other types of machine learning for other models, you would just have to do a massive parameter search, right? You know, and that was always a big computing problem with support vector machines to look at your C's and gammas or whatever.
Have you looked at all into diffusion models, which have largely, to a certain extent, replaced GANs and state-of-the-art models?
I haven't, yeah, for this type of problem, but you know, if you know of a relevant paper, you can send it to me. I'll share it with you know, the postdoc who's doing some of this work. But yeah, I learned about GANs at the Fields Institute, you know, I don't know, must be 5 years ago now, but I was like, I know what I want to use that for. So you know, so yeah, but you know, I mean, time marches on, but at the end of the day, I don't know. I mean, machine learning models come into fashion and go out of fashion. And so, you know, for a while, support vector machines were really popular, and now neural networks are popular, and then maybe now diffusion, but use what works, right? Because, you know, there are problems that are inappropriate for a neural network because you don't have the volume of data you need to really train one. And so you have to kind of go back to the older methods that maybe are not as fashionable anymore, but they're going to give you a better answer. So anyway, I mean, the GAN seems to be working okay with us, and I don't argue with success. So. Questions?
I guess I had a question on the earlier part of your talk. You were talking about careers at national labs and you were talking about like the different tracks. Do people ever move between those tracks, or if you're hired as a staff scientist, do you pretty much like stay as a staff scientist?
I mean, most people pick a track and stay with it. If you pick the technical track I mean, sometimes people maybe will spend a short period of time in the technical track and switch over to the research track. You don't want to stay in the technical track too long because you're not going to be leading the papers, right? And so you do that for too long and then your resume is not going to be compatible with the research track. And occasionally, I mean, very occasionally, you might have somebody on the research track and, you know, they're like, you know, you got some guy that's like, hey, actually, I want to spend more time with my kids. You know, I'd like a 40-hour-a-week job. And not that the research track, they're not as bad as being a professor, but they're probably a little more than 40 hours. But the technical track positions are 40-hour-a-week jobs, right? So, you know, so you get somebody that's like, hey, work-life balance is important to me. And so, you know, you can, you know, we wouldn't, how do I want to say, if somebody were somehow not successful in the research track, it might be an option to them, but it's more likely they would just kind of, you know, move. So, yeah, there are different choices. It's some people refer to it as feeders and eaters. Because the people who generate the grant money are the feeders and the people who do the work on their behalf are eaters. But I don't know if that's, I mean, at the end of the day, everybody contributes in an important way. So I don't know that I see it like that, if that makes sense. Please.
Kind of similar. Do you know like roughly like how many people like stay kind of just at one lab for a long time, and like if people do move, do they usually move to other national labs or do they usually go to like private companies? Just like
All of the above. All of the above. There are people in my division that have been there for 40 years, right? That started their career there and they will retire there, and probably have all their retirement points, but they're not quite, you know, they still have goals and so on. We do have some people that hop a bit between national labs, not, you know, serially, you don't hop and hop, but you know, you can move a couple times in your career between national labs and, you know, build a new program. And we do have, you know, it always hurts a little when I see my early career staff take a job, you know, in industry just because I want them to stay with us. But sometimes, how do I want to say, you know, there are only so many ways to move up. And, you know, there's kind of an advancement track, and sometimes they get a very nice offer from a company that has more leadership potential for them. And, you know, I mean, I hate to see them leave selfishly, but I'm happy for them because we don't have as many leadership opportunities as we have people who are deserving of them. So that does, all of the above happens. And we've had also people that come from industry at a more mid-career level and come to the laboratory. And so we, you know, bring them in as a strategic hire and try to, you know, help them. There, for example, like at least in biosciences, if you go to the NIH, if you've had a career in the biotech industry, but you don't have a record as a principal investigator, there are sort of ways that they can map your background a bit so that you can be competitive for grants. And in terms of our Department of Energy things, we would just try to integrate people into the big programs and then give them the skills they need to go out for more independent funding as well.
Yeah, for the postdocs in the national lab, do they work independently somehow, or they work for a specific staff scientist?
You would definitely be assigned to some projects. Sometimes you might, or maybe a couple projects. If they offer you something where you're split across 4 projects, I would say thanks, but no thanks, right? So, you know, maybe at most 2 projects, 2 PIs. But it's different than, you know, if you get an NSF postdoctoral award, for example, you know, you can go work on whatever you want. And that's not the way the labs work. The labs have projects, there are milestones, and again, unlike, you know, with some granting agencies, you know, you can apply with your ideas to get the grant, but that doesn't always align to what you do. At the national labs, there's more of an alignment between the milestones, right? The milestones and the projects. So you would be, you know, you'd be assigned to a, you know, a project. We also, I guess I forgot to mention and should, at Oak Ridge, we have a distinguished staff fellowship program. And so this is kind of a, if you're really, really good coming out of grad school, you can apply, or after maybe your first postdoc, if that goes well, you can apply. And it's kind of a transition from staff or from like, you know, a high level postdoc into staff position. It's paid as a staff position, but you actually get to write up what you want to do research. You have to make sure, I mean, if you're gonna do that, my advice is get in touch with somebody at the laboratory, someone like me who's a division director. Make sure that whatever you're pitching is actually where they're gonna be hiring down the road, because you get people who, you know, propose wonderful things, but isn't necessarily something that my division can support long term, right? So, but there are opportunities like that as well. And then there are, yeah, and there are other ways to have, you know, a little more independent research, you know, internal funding, so you can do kind of seed proposals. And there's something called lab directed research and development, or LDRD, at national laboratories that are, you know, you make internal pitches for areas, capabilities development, areas development, where you see opportunities in the future with the Department of Energy or other agencies, and can kind of build that up. So it's a little, there's a little less, you know, there's not the complete academic freedom that you have in academia. There's certainly some intellectual freedom, but the, you know, in terms of how you approach the problems, but the types of problems you work on are going to be more prescribed by the needs. You know, these, it's a very mission driven type of research enterprise.
So this depends on the funding. So if it's an external fellowship, so you have more freedom, but it's funded by a specific project, so you have to work on it, right?
Right. Yeah, that's the short answer to the long answer I just gave you.
Another question is, so in working for national lab, is there a restriction of the citizenship? I think
No.
No,
I mean, there will be some countries that, so there's a vetting process, even to interview at the national laboratory. Somebody will check out your background. You know, for certain countries, you know, certain countries have programs that are specifically designed to train people to go and do industrial espionage. And so they will go and they will look for that, right? And so, you know, they'll look for all that kind of stuff. There are certain countries that are what you would call sensitive countries, where maybe we don't have good political relationships, or there's a history of this kind of industrial espionage, and you'll get checked out more. Now, obviously, some people at the national laboratories have security clearances, and that would not be something, you couldn't work in the national defense space as a foreign national, but the type of research that I do in my division, lots of people are from China, India, you know, other places that are producing good folks.
Thank you very much.
Sure.
Follow up with a group discussion. It will be here and not in the room that I advertised. So.
Stop sharing. Zoom. Thank you, everyone, for coming.